Hello, my name is Jordan McKinney, and I am a senior working in the Laboratory of Organismal Biology under Dr. Lauren O'Connell. And my project is on the neural mechanisms of juvenile aggression. I'm interested in juvenile aggression, especially in the realm of different psychiatric disorders that show as displays of aggression in childhood. And in terms of being able to treat these disorders, the neural mechanisms are not well understood. For this project, I'm using Ranitmea imitator, also known as the mimic poison frog. These frogs are biparental and monogamous, and looking across different species of frogs, these tadpoles are a good candidate for aggression because variation in pool size leads to different displays of tadpole aggression. Because of a smaller pool size and increased tadpole care by the parents, these tadpoles tend to be very aggressive with conspecifics, even cannibalistic in defense of their resources. What I want to do is try to determine the neural mechanisms for aggression that cause these displays. Additionally, looking at previous experiments we've done in different brain regions, neural activation differs depending both on the brain region and on the type of behavior, whether it be aggression or control. Neural activation is also shown to be highest in the anterior amygdala. The question that I was trying to answer with my project is looking at what cell types influence aggression in juveniles. In order to determine this, I used a technique called phosphotrap, in which we were able to molecularly profile active neurons in the brain. So first you have your stimulus and controlled tadpoles and behavioral assays. So for the aggression behavioral assay, tadpoles were placed with a smaller stimulus tadpole and left for 30 minutes. For the control trial, the tadpoles were placed with a metal bolt and left for 30 minutes. These were recorded and behavioral tendencies were observed. The brains were then sectioned into three parts: the telencephalon, the diencephalon, and the spinal cord. Phosphorylated ribosomes, which are a marker of active neurons, were immunoprecipitated, which left activated neuron mRNA. RNA sequencing was then used to determine which genes in the brain are active during aggression. Through this, there were several interesting genes implicated, which are labeled in this plot. So there's forkhead fox protein 2, which is a transcription factor involved in communication. It is downregulated. Neurotrimin is also downregulated, and it's been implicated in aggressive traits for children with ADHD. Propiomyelanocortin, also known as POMC, is a precursor polypeptide with multiple post-translational peptides, and it is also upregulated. Alpha MSH, beta endorphin, and ACTH are all examples of POMC peptides. GMP6A is a downregulated gene that's been implicated in panic disorders, and MLC1, which is a membrane protein, is an upregulated gene that's been implicated in fly aggressive traits. As you can see by looking at these plots, there are hundreds of genes that are up and down regulated. We chose the ones that have had prior research done on them that could implicate them in the regulation of juvenile aggression. So we had previously done immunohistochemistry with FOXP2 and PS6, which is a marker of neural activation, and we quantified both the number of FOXP2 cells and PS6 FOXP2 co-localized cells throughout the brain. And this shows us that there's a significant difference between the number of FOXP2 cells in different brain regions during aggression. We then chose to focus on POMC because of its multiple post-translational subunits. Alpha MSH is implicated in feeding and sexual behaviors, ACTH is a mediator of the adrenocortical stress response, and beta endorphins are endogenous opioid neuropeptides. In order to confirm the effect of POMC on aggression, we used fluorescent in situ hybridization in order to selectively tag POMC neurons coupled with a PS6 antibody. By doing this, we saw that there was not a significant difference between POMC cells in aggressive and controlled tadpoles. Next, we did pharmacological manipulations with POMC in order to functionally determine the influence of POMC on aggression. Tadpoles were injected intraperitoneally or intracranially with an alpha MSH antagonist, ACTH agonist, or a beta endorphin antagonist. After a recovery period, the tadpoles were placed in an aggressive counter with a conspecific, and behavior was quantified. This showed us that while tadpoles did not show increased aggression with an alpha MSH antagonist or with injections of ACTH, tadpoles did show increased aggression with beta endorphin antagonist injections. And this is significant because this suggests that the reward system, specifically the mesolimbic dopaminergic pathway of the brain, may be implicated in aggression. In the future, I plan on doing anterograde neuronal tracing in order to look at neural circuitry during aggressive behavior. Because of the implication of the beta endorphin signaling, I would focus this neural tracing around the reward system of the brain, specifically the ventral tegmental area in the midbrain to the ventral striatum of the basal ganglia in the forebrain. These experiments will better help us understand the mechanisms behind juvenile aggression and how to better address the problem of juvenile aggression in society. Thank you.